Snap, we got another caller. Let's go. Hello.
Hi. Um, I wanted to ask about certain parts of aTyr stock.
Okay, I'm here.
So I guess could you explain to someone who's relatively unfamiliar with lung pathophysiology, how exactly do you determine that aTyr's lead doesn't work when you compare it to what was it? Excuse me. It was a TNF alpha inhibitor that you compared it to in your paper. I
Yeah.
didn't really understand like how you analyzed that.
Well, just on that thing specifically, there between the TNF alpha inhibitor and the IL-12 inhibitor that was tried in that trial, there was no efficacy with either of those drugs. And in fact, placebo beat those drugs, which was kind of shocking, numerically at least. And those are between those two drugs, you're covering a huge amount of the autoimmune sort of space. And so I think aTyr management has said that they think their drug works through in some kind of immunotherapeutic perspective, whether, immunology is a big space, but, they think that's how it's quote unquote working. Of course, I don't think it's working at all, but that's their hypothesis, is that it's autoimmune related, that it's doing something in, the immune homeostasis or something like that. And it's really, hard to believe, because if you, one, I mean, the mechanism is kind of out from left field, but two, there had been many trials of autoimmune drugs in sarcoidosis, and they've all failed, including the J&J, those two drugs from J&J, but not just those two drugs, many others as well. Sarcoidosis has been, attacked by Pharma many times, and nothing's really worked. Even steroids don't really do that great in their first line therapy. So sarcoid is like not necessarily that autoimmune, and it's a tough disease to sort of, like I said, there's like been about 20 clinical trial failures for sarcoidosis, and I think that's a bad sign. It certainly doesn't mean that there can't be a first. Of course, there's often that does happen, but in the history of medicine, as far as I know it, if you find a disease like Alzheimer's, where there's just been a litany, or pancreatic cancer even, where there's just been a litany of failures, there's nothing to grab onto therapeutically, and those diseases are often just in sort of tractable. And I feel like going after an intractable disease that's not well understood is often like, hubristic, and kind of a motif that works really well for biotech shorting. Like biotech has been a great shorting space for like Alzheimer's, where again, we don't really understand the disease. We have a couple of clues like beta amyloid, but nothing's really worked. And there's been literally 100 phase 3 failures in Alzheimer's and only 2 successes. That's not a great batting average. And again, I'm not too thrilled with this thesis, because, it's not that causal. It's not really legitimate from a logical perspective, but it's certainly not a positive thing that nobody's been able to ever crack this disease. I think that, is basically playing the game on hard mode. In rheumatoid arthritis, there's like 20 drugs for rheumatoid arthritis, because we understand that disease somewhat, and we know how to kind of tackle it. So I don't think it's positive that, nobody's figured out sarcoidosis. And in fact, the main therapeutic hypothesis has sort of been repudiated by many, many trials. And then I think the bigger part is, how does this drug actually work? Whenever I look at a medicine, I'm really trying to like visualize in my mind's eye the drug entering the bloodstream from the IV, zooming in to micron like level resolution, and asking myself, like, where is this drug going? Where is it going to bind to, and why? And what is it doing exactly? And I think 99% of the people involved in aTyr stock, long or short, can't even answer that question. And it's a fairly complicated molecule. It's this like, it's a Franken protein. It's like FC region plus the synthetase warhead, and they didn't know what or why they were making this. they figured out after the fact that it was that it bound to one of these 4,000 proteins. But I must say, if you put a drug in an assay with 4,000 proteins, you're going to get a hit. And it's kind of like this really backwards logic of designing a drug. Usually you spend all your time thinking about like, okay, what do I want to do biologically? And then you say, okay, well, this is the target I want to attack, and you make a drug to that target, and that's pretty much how 99% of drug discovery works. They kind of did this in this like backward way, and then quote unquote discovered this, neuropilin 2, which had never before been really talked about as having anything to do with sarcoidosis. And again, this does not mean that it's impossible that it has something to do with sarcoidosis, or that it's like the key thing that's been missing. It's possible, but it's very unlikely to find it by chance. And then if you look at what neuropilin actually does, it's not a signaling molecule. And in fact, if you completely get rid of it in the body, there's not really any big changes. You don't have sarcoid, for example. So it's kind of weird that it would be, a drug target. What does it do that makes a difference? And so what it does is it binds, it sort of holds two molecules in place to get them to bind together. It holds, in one hand, it gets a molecule called semaphorin, and then the other hand, it binds VEGF. And VEGF's a really important central actor and could be relevant in sarcoidosis, but blocking or binding to neuropilin, I can't see how that would help or like be an agonist. So it would have to be an antagonist, and so you'd have to show that stopping those two molecules from interacting would somehow be beneficial, and you'd also have to show that it does stop those two molecules from interacting. And I believe they're trying to say that it does not stop those two from acting, which to me begs the question, then why bind it at all? You can only do one of two things. You can activate a receptor, but this receptor has no intracellular signaling. It's just an extracellular receptor. It's kind of inert. It just acts as a co-receptor with those other two proteins. So I don't really understand what it is. And I will say, like, I'm humble enough after 25 years of looking at biotech, a lot of times the company has no clue how the drug works, but it still works. That has happened many times. And you have to be careful that, they just don't know, and that's okay. They're guessing, they're trying different shit, and they might figure it out 10 years from now. So you have to be a little humble and say, okay, what if? And the problem with the what if here is that the data is not good. If the data were good, you would see a dramatic drop in steroid use, and you would see improvement in lung function, dramatic improvement. That's not what we see in the trial results. The results are really modest. There's a no dose response, from 1 mg to 3 to 5. Those are not major dose differences. So there is a dose response. But I question why, for example, the 1 mg dose numerically is worse than placebo. And it seems to me that, like, unless they got the dose response perfect, the 5 mg per kg, which is a big dose for a protein, would not necessarily be like, that's active, but one doesn't work at all. Usually you get a little bit of activity, even at 1 or 3, and then the highest dose does really, really well. And I think there's a lot of like problems with the data. The whole like reducing steroids is very squirrely, because it's like, well, most patients can reduce their steroids while they have sarcoidosis. So if you tell them to stop using their steroids, they will slowly go off of it. And, that makes, I think, running a trial like this really hard. And like, at the end of the day, patients, the disease is not just, okay, I have steroids. The disease is, I have a lung function problem. Because if you just have sarcoidosis and you have no symptoms, you don't need steroids. You only take the steroids where you have a problem and you say, okay, I have a lung problem, I can't breathe that great, I need these steroids, and you take them and your breathing function comes back, hopefully. But then when you stop taking the steroids, a lot of patients can just stop and the disease self-resolves. So I don't know, this thing's like really kind of wild. And I feel like if it was a miracle drug or a good drug even at all, you would have like 100% steroid-free patients in the drug arm and like 50% steroid-free in the placebo, which is what kind of the data has for placebo. But instead, they have like 50 for drug, 48 for 1 mg, 40 or 53 for 3 mgs, and like 58 for 4 mgs, and like that's all the same shit. That's all around 50%. That's not a drug. That's just common sense. A drug is when placebo's at 50% steroid reduction, 1 mg is like 60, 3 mgs is like 75, and 5 mgs is like 100% steroid reduction. And there are other drugs that do that for other diseases, of course. So to me, this is like the weakest drug you've ever seen, even if you could believe that high dose group, and it's a drug where you can't explain how it works. To me, that's always a recipe for disaster. So that's how I think about it.
So let me make sure I'm understanding you correctly in that last part. Did they have different amounts of steroid use for different patient groups? Is that normal or no?
Yeah, so the baseline, obviously each patient has a slightly different, they come into the trial on steroids. That is an inclusion criteria. They have to be on steroids actively right now to get into the trial. And everyone has a slightly different amount of steroids. I think it was 13 milligrams of prednisone on average. But, some patients were taking more, some patients were taking less. And, we feel like the 1 milligram and the placebo groups had sicker people than the 3 mgs and 5 mgs. And you can, I think, see that pretty obviously in the data. And as a result, the sicker patients get worse over time, and the healthier patients stay healthy. And this was not a trial that really was about their drug. This is a random trial of 10 people who have sarcoidosis and are sick, 10 other people who have sarcoidosis and are sick, 10 other people who have sarcoidosis and are not as sick, and then finally 10 more people who have sarcoidosis and are not as sick. And after the trial completed, we saw that the sick people got worse and the healthy people stayed comparatively healthy. And that doesn't tell you anything about the drug, unfortunately, because the baseline was too imbalanced to really read through anything. And the results were not, like I said, like some dramatic home run. They were, like I said, around 50, 48, 53, and 58, which is like all kind of in the same ballpark. That's not like you kind of need a microscope or magnifying glass to tell the difference. So I think that the patients, had their steroids reduced throughout the trial. It was a steroid taper. They had the doctors tell the patients to lower their steroids to, I think, 5 milligrams. And in fact, on the primary endpoint, the number of people who successfully tapered was the highest in placebo. They redefined that when they published their paper by saying, well, we can't show that would be bad. So they instead looked at the total steroid reduction, which is a second way of looking at it, and that's fine. But the way they thought about it before they did the trial was how many people reached the taper goal. And placebo actually had the most amount of taper successes, which is kind of like, that's really scary if you're investing in a drug. You don't want to see that. Any pharma that saw that would say, okay, back to the drawing board. We're not going to develop this anymore. So I think, it's a really hard thing to parse. And the simplest explanation is it just doesn't work. Like the amount of hoops mentally that you have to jump through to believe this drug works, it's just too many. Like it's just more obvious that this drug has no activity, which most small drug companies, they're swimming upstream. This is why I trust Pharma a little bit more, because Pharma has like 40 drugs and in the pipeline, And if one doesn't work, they can be honest about it and just say, if this shit didn't work, why would we spend more money on this? We have 40 more drugs coming next year. While a company like aTyr, or aTyr, they have one drug. So they have to put lipstick on the pig, as they say on Wall Street, and they have to dress it up and do something. Otherwise, why does the company exist? Why are they a publicly traded company? Why should their CEO be getting paid a lot? It's a lot easier to just do the phase 3 and say, well, we're a phase 3 trial company. And if you've ever gone to a compensation committee on a board of directors, I've had the privilege of being on a compensation committee. You hire these compensation consultants, and when it's time to determine your salary for the CEO and CFO, you look at comparable companies. And the comparable companies are other phase 3 biotechs. If you look at comparable companies of phase 1 biotechs or biotechs that have no drugs in the pipeline, the comparable salary estimate is much lower. So these CEOs are often incentivized when they have they have a drug that doesn't really work, to kind of get a little bit more aggressively develop drugs that maybe would be not so developed in other companies.
Okay. This is sort of tangentially relevant to what you were saying earlier. When you mentioned that things like Alzheimer's or sarcoidosis are very difficult to treat, can you then make the statement that you could arguably blindly short companies with a lead product seeking to treat these?
I wouldn't do that. Like I said earlier, I don't like I think a lot of people do that, but I don't. I've been criticized by some of my peers for being too careful and too thorough, because I waste a lot of time looking at every little detail, and that costs me opportunity cost of looking at more stocks, which could provide even more profit. But to me, I like to know that everything about it is bad. The data, the clinical data, the preclinical data, the mechanism, everything. The disease being hard is another, check in the box, but obviously many diseases were hard for a long time, and then somebody cracked them. Cystic fibrosis is a really good example. That disease was basically impossible to treat, and then all of a sudden, there are, it was a huge win for Vertex. So in any event, that was really it. Did I get everything?
Oh, yeah, for that question. Sorry, I have a bunch of follow-ups, but I know you have other callers, I assume.
I don't, but I also want to eat and do other things.
Oh, I'm sorry. All right. Do you have time for one more question?
Yeah, go ahead.
Pretty short. Okay. When you talk about healthier or better performing or worse performing patients in these trials, what is the metric you're looking at? Is it FVC, or is it something about ACE, angiotensin converting enzyme? What are we?
I don't think it would be ACE. All the lung function at the beginning of the study, and then how did they do at the endpoint? And again, the endpoint, there's two ways to explain the endpoint result. The null hypothesis is the result is a reflection of the baseline characteristics. The hypothesis that you're hoping is the answer, if you're the company, is that it's the drug that's making the difference. Nevertheless, there is always going to be some amount of baseline influence that is reflected in the results, which is why you try to, randomize as many patients as you can and nullify that whole large, potential bias. So I believe that. Yeah, I got to take this other caller. Thank you so much. Hello.
Hello, Martin.
Yes.
Oh, yeah. So I just had a general question in regards to someone who's, I'm 32. I got the MBA. I did the finance degree, shit like that. But I never really used it.
I'm only really talking about aTyr right now. I hate to.
Oh, sorry about that. No.
No worries, brother.
Thank